In this activity, you will observe real plant cells undergoing meiosis inside the anther of a lily flower. Unlike our paper models, these are actual reproductive cells preparing to form pollen. Because many cells in the anther divide at the same time, this tissue provides clear, synchronized views of meiotic stages. The anther is the male reproductive structure of the flower. Inside it, special cells undergo meiosis to produce haploid pollen grains. One advantage of using anther tissue is that many cells are dividing at the same time, so you often see clusters of cells all in the same meiotic stage. This makes identifying stages much easier than in tissues where division is scattered. Before you begin observing the anther under the microscope, think about the role the anther plays in the life cycle of a flowering plant. The anther produces pollen, and within each pollen grain are the male gametes that will eventually participate in fertilization. Ask yourself why these cells must be produced through meiosis rather than mitosis. Consider what mitosis does to chromosome number and how it creates genetic variation. If pollen were produced by mitosis, how would that affect the genetic makeup of the next generation? Pause the video and record your prediction in your lab notebook. Then resume when you're ready to begin examining the slide. We begin under low power and locate the anther lobes. These are the chambers where pollen-mothered cells are undergoing meiosis. The photo on the left isn't my photo, but I wanted you to see the overall look of the cross-section. So imagine the anther on the top left cut so you can see the inside. The blue and purple photo is a closer view of one of the lobes. At this magnification, you are orienting yourself and finding the regions where cells are densely packed and actively dividing. When you switch to high power, individual cells and their chromosomes become visible. Because the cells are free-floating at the time the anther is sectioned for slide preparation, the orientation of cells can appear different depending on how the cells were positioned, so some may appear in different meiosis stages, but they are all in the same stage. Since these cells are in the process of division, the DNA is condensed and stained, allowing you to see chromosome shapes, alignment, and separation. Here are different slides of anthers in various stages. How can you tell what stage of meiosis it is in? Examine the arrangement of chromosomes carefully. Are homologous chromosomes paired? Are tetrads visible? Are chromosomes aligned at the center, separating, or forming new nuclei? Use these visual clues to decide whether the cells are in prophase 1, metaphase 1, anaphase 1, or one of the stages of meiosis 2. In the top left video, all the cells show condensed chromosomes in each cell. This was identified as late prophase 1 since they are starting to move to the center. The top right image shows 1, 2, or 4 cells with chromosomes at the opposite side of the cell. This would be anaphase 2. So why wouldn't the ones with 2 cells not be anaphase 1? In anther tissue, groups of pollen mother cells often enter meiosis at the same time. This synchrony means large clusters of cells progress through the same stages together, making it easier to identify patterns of chromosome behavior. When the pollen mother cell was cut, it may be positioned where half of the cell was cut, so you can't see the other half of the cell going through meiosis. So if you see 4 cells forming anywhere in that view, then you are in meiosis 2. The cells in the bottom photo are in what stage? If you said telophase 2, then you would be right. Since a couple of the cells show 4 cells forming cell plates with chromosomes in their nuclear membrane, that would indicate telophase 2. Here's one of the images I took. Based on what you learned in the previous slide, what phase would this be in? Draw a couple of pollen mother cells and label it with the phase you identified. Pause the video to do your sketch. Look for key indicators such as pairing of homologous chromosomes, separation of chromatids, or formation of new nuclei. Pause the video and record your reasoning in your lab notebook. Consider how a 3-dimensional process is being viewed in a 2-dimensional slice. Some structures may be partially cut, hidden, or distorted. Pause and explain how this affects what conclusions you can confidently draw. Plant reproduction has so many unusual cases that plant breeders take advantage of. Most plant cells are diploid, meaning they contain two sets of chromosomes, one from each parent. However, plants often show an unusual condition called polyploidy, in which cells contain more than two complete sets of chromosomes. For example, a triploid cell has 3 sets, a tetraploid has 4 sets, and so on. Polyploidy can occur when chromosomes fail to separate properly during cell division, or when two species hybridize and their chromosome sets combine. Unlike in animals, polyploidy is very common in plants and is often not harmful. In fact, it can be beneficial. Polyploid plants frequently have larger cells, thicker leaves, bigger flowers, and sometimes greater tolerance to environmental stress. Many familiar crops are polyploid, including wheat, strawberries, bananas, and potatoes. The large size and seedless nature of some fruits are direct results of polyploidy, like seedless watermelons and grapes are triploid. From an evolutionary perspective, polyploidy is one way new plant species can arise quickly. A polyploid plant may become reproductively isolated from its diploid ancestors, allowing it to follow its own evolutionary path. This is another example of how changes in chromosome number, whether through meiosis, mitosis errors, or polyploidy, can have visible effects on plant structure, growth, and diversity. This activity shows that meiosis takes place in the anthers where pollen is formed. The cells you observed are undergoing meiosis to produce haploid cells that will eventually become male gametes. Because the chromosomes are condensed and stained, you can directly see their arrangement and movement during different stages of division, allowing you to identify specific phases of meiosis. Many of the cells in the anther divide at the same time, which means large groups of cells are often in the same stage. This synchronous division makes it easier to recognize and compare chromosome patterns. Finally, the way the anther tissue was sliced to make the slide affects what the chromosomes look like under a microscope. Depending on the angle of the section, chromosomes may appear aligned, separated, or overlapping, even if they're in the same stage of meiosis.